So why don't we go ahead and get going with Scott and his talk. It's a pleasure to welcome Dr. Scott Coffin from the California OEHHA program, the Office of Environmental Health Hazard Assessment. He is going to be giving us a talk titled Towards Understanding of Exposure, Hazard, and Risks of Microplastics. Scott, take it away.
Awesome. Thanks, Matt. And it's great to be here with you all for coffee. Somebody asked me on social media if the coffee and microplastics webinar was going to be about the health effects of coffee. And I said I would never do that kind of research because I'm not going to ruin the one thing that brings me joy absolutely every morning. So,
Let's make
anyway,
a pact. Let's all agree to do that.
yeah, that's a pact. So I'm going to talk a little bit about why California has been working on microplastics for past 6 or 7 years, and then jump into how we've been assessing the human health hazards and how the ecological hazards are assessed a little bit differently. And then I'll cover a little bit of the ecological risk characterization that we've done in San Francisco Bay and that we're currently conducting in Monterey Bay, and showing you how that process has evolved over time. Talk about databases, Tomex2, as well as some funky AI and ML applications, and then end with quality standards and open science. So spanning a lot of topics today. Hopefully I don't go a little bit too fast for y'all, but I'm happy to share the slides afterwards as well. So California Senate Bill 1263 is one of the main reasons that I've been working on microplastics. This bill was passed in 2018 and compels the California Ocean Protection Council and state water boards to initiate a statewide microplastics monitoring and mitigation strategy. And included in that is coming up with a risk assessment framework so that we can prioritize the ecosystems that are at most risk. We also had a similar bill passed in the same year that required the State Water Board to address microplastics specifically in drinking water, starting with a definition and then coming up with a plan for 4 years of testing and analysis. And I'm not going to cover this in super depth as I no longer work at the State Water Board. It's been a couple of years, but I was in charge of this from 2019 to 2024. Currently, what I'm working on is California Senate Bill 1147. This was passed in 2024 and requires OEHHA, my agency, to develop a assessment, a human health risk assessment of microplastics in both drinking and in bottled water. And the goal of this bill is to come up with a standard that could then be used for monitoring and potentially regulations for microplastics in drinking and bottled water. So this is basically a reboot of that 2018 bill. We also have a really exciting bill that was passed in 2022 that is a comprehensive packaging extended producer responsibility bill, also known as SB 54. And this bill requires manufacturers of plastic to ensure that their material is actually composted or recycled. And if it's not if it's not meeting certain targets by certain years, then they get slapped with a fine. So really closing the loop here and putting the onus on the producers to really make sure that the whole life cycle works of capture and recycling and all that. And notably, it includes $5 billion paid by industry over the next 10 years to mitigate the impacts of plastic pollution to disadvantaged communities and Californians at broad. So every year we get $500 million. 60% of that goes towards environmental justice and public health mitigation, and 40% goes to environmental plastic impact mitigation. And so this is a really exciting opportunity for California to have sort of like pie in the sky ideas and address plastic pollution with a decent amount of funding. And so we're just going to be getting started with this next year. Another exciting thing that's happening in California, just recently, the California Department of Toxic Substances Control, or DTSC, added microplastics to the candidate chemicals list of the Safer Consumer Products Program. And basically what this is a green chemistry program where the agency looks at chemicals or contaminants in products that pose some type of exposure or hazard or risk to consumers or ecosystems. And they effectively go through an alternatives analysis where they consider what other materials could this be made of, what other chemicals could be included that would be safer or more sustainable. And the producers have to produce their own report where they basically um, um, put together a proposal of like, okay, we're going to change it to this, and then DTSC works with them to make sure that they're taking the steps to ensure that they're not doing any regrettable substitutions. So it's a really exciting thing. It's one of the only source control measures specifically targeting microplastics at broad anywhere, and it's happening right here in California, which is really exciting. So now into more of the science, human health hazard assessment. We had a workshop from 2020 to 2022. It's pretty outdated now, but a lot of the information with regards to the framework that we came up with is still pretty valid. And I'll talk a little bit about how it's evolving and where it will likely go. Part of this workshop, we compelled or we asked the experts to consider which dose metrics cause the toxicity of microplastics. Is it the particle shape, size, surface area, mass, particle count, what have you? What type of particle characteristics make these microplastics more or less toxic? And what are the adverse impacts to different organisms? Are there adverse outcome pathways that we could establish? And can we fold all of this into a threshold framework that could then be used to inform management? As everyone in this group probably knows by now, microplastics are a very broad contaminant suite that causes toxicity through particle, biological, and chemical transfer means. The physical effects are different for organisms depending on how big they are. So really small critters could be entangled by very small microplastics, like C. daphnia. Fish and other organisms that eat larger plastics can suffer internal lacerations or food dilution. And for humans, we mostly care about the cellular impacts of really small microplastics causing inflammation and oxidative stress. At the time, there were really few studies. This is a pretty old systematic literature review, but back in 2022, there were only 29 ingestion-based in vivo mammalian microplastics tox studies. We screen these with custom quality criteria to make sure that the exposure and hazard data was being reported well, and then filtered this down to 12 studies that were considered fit for purpose. Most of the studies were focused on male reproductive impacts, and as we all know in the field, we've been looking at a lot more impacts since then, a lot of more endpoints. But this is kind of the quantitative dose response assessment that we did back then. You can see here that the point of departures by mass don't range dramatically, even though we have particle sizes from 40 nanometers up to 20 microns. So back at the time, we had a very small database to work with. We did not feel great about using this, and so we decided that we were not going to release any quantitative numbers that could be used for a regulation, largely due to this inadequate effects database, uncertain effect mechanisms, and the incomplete exposure database that we're still trying to resolve. So 4 years ago, where has the field gone? As I mentioned before, we've got these particle, biological, chemical impacts. Most of the studies that we see in the field are testing the particle-based impacts. They're using pristine polystyrene spheres. Not very representative, but it is a simple thing to use. It removes some factors. The pathogenic impacts has been studied a little bit, but we think that there's a lot more that needs to happen here. And the chemical transfer, there's been a lot of modeling studies that have been done, and it's looking like it may be a relevant factor, but there's still some more research that needs to be done with this, in my opinion. And this is primarily because there's just too many chemicals to comprehensively understand and model with these plastics. We know that there's over 10,000 additives, and a lot of these are hazardous. And the particles, once they enter the environment, they're transforming. The chemicals on the particles are leaching or becoming oxidized and undergoing environmental biological transformation. These particles also gain eco-coronas, microbial colonization, a lot of different impacts that can change their biological uptake, how long they stay in the body, and what type of additive transfer they may present to the organisms that ingest them. Dr. Coleman's group from Wageningen in the Netherlands has released a number of PBPK models or pharmacokinetic models. And they just released an update. I'm not showing this here, but just about a couple weeks ago. And this is really where the field needs to be going to get a quantitative understanding of how these particles are moving through a human body and which organs they are residing in, for how long. And this will be useful for when we get quantitative, like really reliable quantitative biomonitoring data, so that we can pair that data with exposure characterization and really have a better understanding of what we're dealing with for these particles. We also are witnessing that there are risk assessment frameworks out there for human health that are evolving over time and converging onto certain traits that we seem to our expert panel back in 2022 put out as something that was important. So this was a paper that I just published with Bart Coleman's earlier this year, looking at all the different human health risk assessment frameworks for microplastics. And we color coded these by whether these frameworks had or met certain criteria, like external exposure assessment, which pretty much all of them did. Very few of these are using this internal biodistribution PBK. It's difficult to do, but this is something where the field is going. And one thing that few of these studies have done is really consider the polydispersity or the plastic continuum aspect, which is a very difficult thing to do, and I'll cover a little bit going forward. Also, few of these studies are really considering the full extent of the quality assurance and quality control limitations, which again is something that the field has struggled with for a long time. Quantitative hazard effect, quantitative risk. These are some other aspects that some of these do well. And then the regulatory alignment is one thing that only our study in 2022 was really geared towards. But it's our next assessment from OEHHA will definitely try and be aligned with that. So my lab is doing some research on microplastics to fill some of these exposure data gaps for our hazard assessment. And I'll talk a little bit about what we're doing. In brief, we're going to be characterizing exposure through bottled and drinking water, performing a systematic literature review, and then we'll also be holding an expert panel to update our risk assessment framework and pour over this literature and data that we put together and come up with a hazard characterization. So we're going to be doing a little bit of nanoplastic characterization using OPTIR for 1 liter grab samples of tap water around the state, as well as bottled water samples, excuse me, with our collaborators, Dr. Bejan Yan out at Columbia University. We're also going to be looking for the larger microplastics, so 1 micron and up, using the state's standardized sampling method released by the Southern California Coastal Water Research Project, or SWRP. This is an inline filtration method that can sample large volumes of 50 to 500 liters. And this will be going through the State Water Board's standardized analytical method for micro-Raman spectroscopy. We'll also be looking at the leachates from bottled water specifically. So we're going to take common bottles that more popular ones that people drink, and basically we're going to grind them up under 2 conditions. One is just pristine, and the other is like a simulated weathering and usage, putting it in the sun, leaving it in your car, opening the cap like you would, and then we're going to take these particles and figure out which chemicals are on them and are they bioaccessible at the particle sizes that people are ingesting them as. So we'll be doing this through a strong solvent extraction, as well as an in vitro transwell migration assay, and then doing targeted and non-targeted analytical chemistry to see what's in these bottled water microplastics. Let's see how we're doing in time. Not a whole lot left. I think I'm gonna, yeah, I'll go through the eco talks and then cruise to the quality assessment, because I want to make sure I cover that. So I mentioned before considering the polydispersity or the continuum of microplastics is something that the field is not always great at. It's a difficult thing to do, and this is the way that we have addressed this for ecological risk. In terms of impacts to macroorganisms, we think about food dilution as something that is really a very understandable and easy thing to model and witness. Basically, plastic's not food. Some organisms, like birds, eat plastic that is bigger than what they can ingest, and so their guts accumulate with plastic over their lifetime, causing them to starve. And we can model the toxicity of these plastics based on their volume, how much space they take in the organism's gut, really and restricting the particle sizes to those that are accessible, so stuff that can fit in their mouth. And then for human health and really all organisms, we care about these tissue translocation impacts. So this is the particles that are small enough to get lodged in tissue, biodistribute. And here we care about the surface area as well as the specific surface area or the aspect ratio. And these are based on nanoparticle toxicity literature. We believe these exposure metrics to be predictive of those types of toxicity. So using that framework, we can use studies that all use different particle sizes, shapes, polymers for different organisms and align them all to one common distribution so that we can come up with one value for a group of particles as opposed to trying to split a bunch of hazard assessments 1,000 different ways. And the way that we've been doing this has been updated a bit. I just released a paper earlier this year releasing what's called a probabilistic species sensitivity distribution plus plus, based on a previous model that would really collapsed or considered the distribution of toxicity across studies. A species sensitivity distribution, for those that are not ecotoxicologists, is what the EPA uses to assess hazard of contaminants, and it's basically a dose response curve of the different species. The issue with microplastics, though, is that you have a lot of variability between these studies. Species have different sensitivities, different endpoints, and a lot of the previous SSD method didn't really capture that variability. So that's what this new model does. Really considers the particle variability as well as the interspecies variability. It's available as an R package. You can download it from GitHub. I'm going to actually cruise ahead past the ecotox risk characterization because I really want to cover the quality assessment stuff, which I think is pretty important. So you just have to catch another talk in order to see what we're doing with ecotox. So I mentioned earlier that we've been collecting a lot of data. The toxicity of microplastics Explore is a living database that also comes with a interactive visualization and data portal. It's based on rShiny. You can find it online at microplastics.swerp.org. This database is meant to be a living database that updates with the literature. And we have been working in a sort of manual stage to get this updated in the past. We had 67 contributors from 14 countries volunteer to mine data for it a couple of years ago. And this is now live. We more than doubled the database size for ecological hazards and nudged the human health database up from 55 to 77 studies. This convergence of data and keeping it all in one place, I think, is really important so that we are not duplicating each other's efforts and we can all work from like a common understanding of what's out there and build upon each other's work, especially as the field is absolutely exploding. It's estimated that there'll be 3,000 microplastic studies published in 2026, if this exponent holds true. We'll see about that, but there's certainly a fire hose of studies, as we all know. And one of the ways that we can deal with this abundance of studies is by leveraging some large language models. Our Tomex 2 effort, we were using a very primitive version of ChatGPT 3.5 without a whole lot of prompt engineering to see if we can extract some of this data from studies using large language models. The results were extremely mixed. Some criteria it was getting really well, others it was absolutely bombing. It was a small subset of studies, and we honestly didn't really know what we were doing. And so we're trying again, this time working with experts in AI out at the Oregon State University. And what we're doing is developing a fully automated agentic pipeline workflow that works on a cron job. So every week or so, however frequency we want to set, it will search the literature and find studies that it thinks are relevant, make sure it's not already in our database, get the full text, and then through a large language model prompt engineering, it will extract different fields and do quality scoring, and then it will have a confidence check. So some things it will have an intuition that will bake into it that says whether it needs to be flagged by a human or not, and we'll know based on our experimentation how well it does at certain criteria. If it fails often, then we want to check those. And then once everything's good, human review, then we'll update Tomex. So this could be something that we'll just update effectively automatically. And we're about halfway through this project, doing what's called a self-evolution and having the model teach itself so that we're not just constantly trying to tweak prompt engineering, tweak prompts. We're also using machine learning to predict the effects of microplastics. This is a paper that was just submitted. Yeah, I'm trying to predict whether a binary effect is happening based on the Tomex2 database. Getting pretty good models. And it's really important that we use high quality data. We find that when we filter the data to only the high quality studies, we get much better modeling results. We can also, I've also been working on a quantitative predictive model with the understanding that the alignments that I mentioned earlier don't fully capture some of the qualitative differences in toxicity between particles. Fibers tend to be more toxic than non-fibers, even when you account for the surface area and volume and all of that. This is a study that came out 2026, looking at the Tomex database that found that. And so really, this is sort of like an add-on, would be an add-on to the PSSD method. And it's a little bit aspirational, but in effect, basically the thesis of this paper is that there's over 280 ecotoxicity studies on microplastics in over 100 species in water, and they're mostly focused on spheres and fragments. And current risk assessment methods can largely account for those differences due to particle size, shape, and polymer using these ecologically relevant metric-based alignments. However, differences in toxicity remain even after aligning for the data, which limits our ability to reliably assess the risks of environmentally realistic mixtures, which contain greater proportions of fibers or transformed particles, weathered particles, than the toxicity database contains. Therefore, can we use machine learning to predict these chronic NOACs across all species to an environmentally realistic mixture of microplastics to construct a more reliable PSSD or hazard assessment? And what I'm finding is that the models are pretty good. There are a lot of intricacies with this work, and data leakage is a major issue. Some other studies have attempted this and have not considered the data leakage, which can lead to inflated confidence in the model. I'm finding that the data is not sufficient right now to really build a robust enough model to see a difference in terms of these prediction capabilities, but I'll be publishing this soon so people can look at it. And I think I don't have time to talk about that thing, so I'm going to skip ahead and just talk a little bit about quality standards and then pass it over to Genoa. So in our 2026 Tomex2 report, one of the studies, we found that a lot of these studies are not reporting certain quality criteria. Over 80% of these studies didn't meet the minimum quality criteria for risk assessment, which in my opinion is a shame. It's really a lack of reporting that a lot of these studies have failed to do. So that's something that is a fairly easy thing to fix. And based on a conversation that I had with different researchers at the UNM conference back in January, we came up with a a paper to provide the field with a sort of a roadmap to the landscape of all the different quality criteria, standards, what have you, so that when someone is designing a study, they know which papers to read or at least where to look, because it's a large landscape, and giving a roadmap, especially to newer researchers, will be really helpful. And so depending on the type of experimentation you're doing, this is for monitoring, say if you're in drinking water, whatever stage you're at has more authoritative documentation or standards than others. Human tissue has basically nothing, just supporting studies, sometimes some critical guidance, but nothing like an accredited method by a governmental institution like for drinking water. So that's a roadmap that we put together. There's also a cool Streamlit app, which is live. You can check this out. This is actually live updating. So as new papers come online, I'm continuing to add them to this. And this is a way that you can go through a different workflow, whether it be monitoring, hazard assessment, risk assessment, and find the relevant studies that are useful for whatever stage you're thinking about for your experimentation or for reporting your results. So yeah, check it out. I can also drop the link in the chat to make it a little easier. And yeah, I'm just gonna pass it over to Genoa because I think I'm out of time. But yeah, I just want to thank all the volunteers and everyone that's helped me with these years of research. And hopefully we'll have a little bit of time for questions at the end, but I think I might have went over a little bit. So thanks everybody.
All right. Thanks, Scott. That was great. I think we do have time for one quick question, but I would also encourage people to jump in the chat, and you've got 30 minutes to converse in the chat while Jenna is going. Are there any immediate questions? Scott, I would ask, what are you doing to address sort of the gap of information related to the nanoscale materials that most technologies don't capture well?
In terms of the characterization that we're doing,
In terms of
or
incorporating that into risk models, like it's the dark matter, right? So are you dependent on stuff that's been reported, or are you sort of extrapolating to what is likely or possibly there?
great question. A lot of these toxicity studies for human health focus on the nano range
Yeah,
and trying to we've ignored them for ecotox because we just don't have exposure data.
right.
And that's something where we are going to fill that gap a little bit with our nanoplastic characterization study of what people are actually drinking in tap and bottled water in California. It's just one tiny piece of the puzzle. We're not going to know how much they're breathing and eating and all of that. So it will be challenging to extrapolate. But what we're finding is that the extrapolation, when you think about these alignments and rescaling, is extremely uncertain. Especially the smaller you go, your error bars can balloon dramatically. Even with a couple, from 10 microns to 1 micron, you're increasing your uncertainty tenfold. And so we really need to focus on getting good, clean monitoring data to feel reliable about these extrapolations.
Right. Okay. That sounds cool. That's good. Jump in the chat box and let's switch over.